were on this page, and I made a lot of promises to you guys. I promised I'd show you the Compare Region Viewer. I didn't promise to show you the Genome Browser tab, which would show you the annotation in BV-BRC and the RefSeq annotation if there were any of them. I didn't promise to show you the Domains and Motifs tab, which would show you if there were any domains that were called on this protein, which doesn't appear to be any. I said we'd look at the Compare Region Viewer. So let's click on this. Now let me warn you, this view is a little shy, and sometimes it takes a while to load. We're going to have to wait for it. And sometimes, after a while of waiting, it doesn't seem to be loading at all. So when that happens, do a hard refresh of the page, which is Control-Shift-R. But look, we were patient and we were able to see it. And so I'm sure that you're looking at this, and if you've never seen this before, you're thinking, what is this? There are colors, there are arrows, there are boxes, there are lines. I don't understand any of it. Don't worry. You're in my capable hands, and I'm going to help you understand it. But the first thing is I want to give you the anatomy of this viewer so you understand that. So you'll notice that there are these red arrows and red cylinders right down the middle. When you use the Compare Region Viewer, the first thing it's doing is it's blasting that particular gene against all the data in BV-BRC, trying to find the closest homologs. And notice up here under genomes, it's looking across the reference and representative genomes. These are GenBank assignments of high-quality genomes that represent what is known about the taxonomy of the bacterial organisms right now. So it's looking across that set, and it's ranking these things based on their blast hit to that gene. So here is the gene, and when I mouse over it, you can see what it is, and it is the same gene as this because we're on this page. Underneath it is the blast hit. You'll notice this cylinder expands across the length of that gene. The first one it is blasting to itself, and so it sees itself. And it's a strong red box that indicates a really high quality blast hit. If you go down a bit, you can see that some of these have a lesser blast hit, and so it's a lesser intense color of red. Some of the arrows are small, and there's a teeny little blast hit. That's saying that this had a pretty good blast hit when you blasted that whole gene, but it's against this tiny little gene in this guy, and this shows the length of the blast hit. Well, while we're down here, what does this mean when you have this here and here? This is an indication that this gene is on a single contig, and there are no other genes on it. But you'll notice that everybody else has all this other stuff going on. What does this mean? So when it gets the best blast hit, and it's ranking these things right now from the strongest to the weakest of these 10 genomes, depending upon the blast hit, it brings in then the genes upstream, which are up here, and the genes downstream that are here. And it's showing if they're red on the forward strand or the reverse strand. So here they all are. Now, if the genes or the proteins are the same color and they have this little number over it, it's an indication that they are the same protein family. They have the same protein family assignment. And because right now we're looking at the cross genus families, this is the PG fam. And if I mouse over this, you can see from this information that this is a mobile element protein. It shows its location, and we would like it to show the protein family ID, but if I were to double click on this, I could see it. I don't want to do that right now, because if you double click on it, it'll redraw the structure based on that. So you can see that some of them have similar upstream regions. These guys have very strong similar upstream regions, the Shewanella and Klebsiella genomes. But when you get to the downstream region, it's something different. And now that we're there, you notice that some of these things are arrows and some of these things are boxes. You can figure out what anything is by mousing over it. I mouse over this arrow and it tells me that it's an aminoglycoside 6-phosphotransferase. Okay? But what about this box? I mouse over that and it's a repeat region. Any protein encoding gene from bacteria in the resource will have this kind of view available. But you can't do it for non-protein coding genes, but you can see them here by mousing over it. This is another different annotation. It says that there is a repeat region annotated there. So in the next video, we're going to talk about these controls to the system so we can show you how you can change the view and the things you see. Come back for this one. It's an important one. All right, thanks a lot. Bye.